In this video, we will discuss about the media preparations in the microbiology laboratory. We will discuss all the steps that we are applying for preparation of water media in the microbiological lab. This one is the overview of media preparation, in which we will follow the different steps, starting from the weighing of culture media, then we'll also go for pH meter and for pH of the media. This one's also very important. Then we'll go for the boiling for complete mixing of the media. The next also is for the sterilizations of some media. For some media, boiling is only for use as a sterilization purposes. After sterilizations, we'll simply do some kinds of plating in which we will pour the media in the plates, and the last we'll go for sterility. In this way, the step by step we are doing weighing of media and pH check, boiling, autoclaves, and plating, and the last we'll go for the sterility testing of the media. Either the prepared batch is sterile and either we can use it or not. This one from the first steps, the most important thing here is we use distilled water, and for ISO, International Organization for Standardization, recommended we need to check the conductivity and total plate count for this media, for this water, distilled water that we are using for our media. This one is very important thing. We need to check its conductivity. What standards say we need to use that media. Next is the culture media. This one is discussed about the here we will discuss about the powder form culture media, in which we need to prepare in the laboratory, but in the market some other media is also available that is ready to use. We will use them also. But here we will discuss about the powder form media, in which whenever we will purchase any media, what we need to do, we need to check its certificate of analysis, either the batch, everything is clear, whatever is it's written on the description on the media bottle, and either it is traceable to the international system, and also we'll go for its quality check. That is also recommended by our ISO 11133. We also need to check the culture media, whatever we want to use in our laboratory. The third step is the weighing. This one is very important. We know each and every component is recommended in the ISO methods. Thus, we need to make a proper calculation by checking the label, what description, how much gram is they simply say to use. We need to proper calculate, and a proper volume of water we need to add, so that agar's concentration will be the same, so that it will become a good for agar to solidify in the plates. In the next steps, after weighing, we will go for mixing. And the mixing, we will simply mix our culture media by using some kind of mixer, stirrer. And the next, we will go for the pH meter. For the pH is recommended, is also written on the bottle. They will simply say this one is the guideline, this one is the limits for pH. pH check. For pH meters, before using, we also need to calibrate the same way for weighing balance. We also need to first verify this one. And for pH meter, we need to calibrate according to our which buffers we are using. We need to calibrate this one, then we go for our pH of our media. If there's any kinds of fluctuation, we also need to add some kinds of sodium hydroxide solution, some kinds of HCl, hydrochloric acids. pH is very important because all our colony colors, colony morphology, colony color appearance depends upon the pH. If pH is not according to the limits what they suggested in our media, I think the bacteria will not grow there properly. If basic kinds of bacteria will grow, want to grow at the pH of the media is acidic, it will not grow properly, will not give a good results by if we will not adjust and check the pH of the media. Next step is the boiling. Boiling as simply instruction as per instructions in some way. Boiling of media is recommended as for the purpose of its sterilization. Boiling is very important. Somewhere it's also as recommended for mixing of the media, proper mixing of powder in the water. This one it depends upon what instruction is recommended by the manufacturers. It's already written on the bottle. After boiling, we'll go for the sterilization, and in which sterilization will go for autoclave. The temperature, its time, its pressure, all these things are very important, especially for sterilization purposes. We need to check, and also autoclave check is very important. Its cleaning, its water level, and its water distilled water. We need to change its. We also need to use the distilled water every time so that our autoclave will run for a longer time. We also need to check the sterilization cycle is completed accordingly or not. We will in the next slides. After autoclave, we also need to check the autoclave verification. This step is a 6 steps. We will use some kinds of strain indicators. This one is a tape. We also use another indicators. We use as ampule. All these items, we just need to check our cycle is completed. Is it according to as autoclave is done properly or not? This one is actually a autoclave verification. No, the media is wild. We simply go for its go to the water bath in which we will. What I also recommend to simply to 44 to 47 degrees. They will simply say we need to add this media in a water bath having 47-degree temperature, and we will place in the water bath until we go for plating because water the media cooling is very important. Otherwise, there is a lot of moisture in our plates if we go for direct plating. The next, as we are going for plating, and for that plating, we need a cabinets and cabinet sterilization, cleaning with proper disinfectants. Either we are using ethanol, 70% ethanol, isopropanol. We also need to switch on the UV light for some for 15 to 30 minutes, whatever recommended by your manufacturers or what guidelines. We need to also pre-clean it before pouring our media. After pre-cleaning, after making a safety cabinet ready, we will simply take media from water bath and place in the cabinet, and we simply place in our we'll place the media in our petri dishes. And after allow it for solidification after 10 maximum 10 to 15 minutes, within 5 to 10 minutes, and somewhere it's longer time depends upon the conditions, it will become solidified. After solidification, there is another step is called as a sterility test. This one is very important. Either what batch you prepared is sterile or not. We need to place in the incubator for 24 hours. All other plates we will place in the refrigerator. We will check its by incubator. Thank you so much for your time. This one is from for the today. Thank you.